Hello students, today I am going to teach you the thyroid hormone. Thyroid hormone is synthesized and secreted by thyroid gland, a large unpaired endocrine gland positioned anteriorly in the neck, below and lateral to the thyroid cartilage. The gland name is derived from its topographic relationship to the laryngeal cartilage, whose shape resembles a Greek shield or Theodorus. The principal hormone secreted by thyroid is thyroxine or tetraiodothyronine, also known as T4, and triiodothyronine, that is T3. T3 is more active than T4 and is also formed in the peripheral tissues by deiodination of T4. A small amount of reverse T3 is also formed, which is inactive. So this is the structure of T4, T3 and reverse T3. T3 is formed by 5' deiodination of T4 in peripheral tissues, but when there is deiodination at the 5th position, reverse T3 is produced, which is inactive. Thyroid gland consists of two lateral lobes with an isthmus that connects the lateral lobes, and this isthmus crosses the anterior surface of the second and third tracheal cartilage. Sometimes there is a pyramidal lobe arising from isthmus in front of the larynx. The gland is well vascularized and thyroid has one of the highest rates of blood flow per gram of tissues of any organ in the body. This is the posterior view of thyroid gland. The parathyroid glands are usually embedded in the fibrous capsule on the posterior surface of the thyroid gland, two in the superior poles and two in the inferior poles of thyroid. Thyroid gland is located anteriorly to the trachea between the cricoid cartilage and supraesternal notch. The isthmus lies anterior to the second and third tracheal rings. The normal thyroid is 15 to 25 gram in weight, highly vascular and soft in consistency. The four parathyroid glands produce parathyroid hormone, located posterior to the each pole of thyroid. The minimum daily iodine intake that will maintain the normal thyroid function is 150 microgram in adults. Out of this 150 microgram, thyroid secretes 80 microgram per day of iodine in T3 and T4. So this is the histological picture of thyroid. Thyroid gland is composed of multiple follicles or acini. Each spherical follicle is surrounded by a single layer of polarized epithelial cells and filled with pink staining proteinaceous material called colloid. Colloid consists predominantly of the glycoprotein thyroglobulin. When the gland is inactive, the colloid is abundant, the follicles are large, and the cells lining these follicles are flat. When the gland becomes active, the follicles are small, the cells are cuboid or columnar, and areas where the colloid is being actively reabsorbed into the thyrocytes are visible as reabsorption lacunae. So the thyroid gland synthesizes thyroid hormone and calcitonin through two distinct cell types. One is the epithelial or follicular cells that synthesizes thyroid hormones, and the other one is parafollicular cells which synthesizes calcitonin. The adult human thyroid secretes on average 94 to 110 microgram of T4 and 10 to 22 microgram of T3 daily, and the major regulator of this process is TSH. So the thyroid cells have three functions. It collects and transports iodine from circulation. It synthesizes thyroglobulin and secretes it into the colloid. It removes the thyroid hormone from thyroglobulin and secretes them into the circulation. So let's discuss these functions one by one. So first is collection and transport of iodine from circulation. So the basolateral membrane of thyrocytes facing the capillaries contains sodium iodide symphoter that transports 2 sodium and 1 iodine into cells with each cycle against the electrochemical gradient for iodine. This sodium iodide symphoter is capable of producing intracellular iodide concentrations that are 20 to 40 times as great as the concentrations in plasma through secondary active transport. This secondary active transport gets energy by sodium potassium ATPase which transports sodium out of the thyroid cells. TSH induces both expression and retention of sodium iodide symphoter in the basolateral membrane. So this iodide, which is now located in thyrocytes, exits the thyrocyte across the apical membrane to access the colloid by a chloride iodide exchanger, which is also known as pendrine. This protein is the product of the gene responsible for Pendred syndrome, which manifests as thyroid dysfunction and deafness. So the first essential step in the formation of thyroid hormone is conversion of iodide ion to an oxidized form of iodine, that is nascent iodine, which is capable of combining directly with tyrosine. This oxidation of iodine is promoted by the enzyme thyroperoxidase. So this oxidized iodine or nascent iodine will bind directly but slowly with tyrosine. But in thyrocytes, the oxidized iodine is associated with thyroid peroxidase enzyme that causes the process to occur within seconds or minutes. Then during the next few minutes, or hours or even in days, more and more of the iodotyrosin residues become coupled with one another, forming iodothyronins. So thyroglobulin is packaged extremely efficiently into the follicular lumen at high concentration. Thyroglobulin is a glycoprotein. It contains 123 tyrosine residues, but only 4 to 8 out of these are normally incorporated into thyroid hormones. Thyroglobulin is synthesized in the thyroid cells and secreted into the colloid by exocytosis of granules that also contain thyroid peroxidase, the enzyme that catalyzes the oxidation of iodine and its binding to tyrosine. Thyroperoxidase is a heme containing glycoprotein located in the apical membrane of the follicular cell. Thyroid peroxidase is involved both in iodination of tyrosine as well as coupling of iodotyrosins. So the binding of iodine with the tyrosine residue in thyroglobulin molecule is called organification of the thyroglobulin. After organification, thyroglobulin must be returned to the follicular cell for further processing before secretion. This occurs by a complex but well characterized process of vesicle mediated endocytosis, which is regulated by TSH. Thyroglobulin is proteolized and thyroid hormone is generated in lysosomes. And these thyroid hormones are secreted into the capillaries on basolateral side of thyrocytes. The major hormonal product of the coupling reaction is thyroxine, that is tetraiodothyronine. In the normal human thyroid, the average distribution of iodinated compounds is 3% monoiodotyrosine, 33% diiodotyrosine, 35% tetraiodotyrosine or T4 and 7% of triiodothyronine, that is T3. Only traces of reverse T3 are present in normal human thyroid. So the peptide bonds between the iodinated tyrosine residues and the thyroglobulin are broken by proteases in the lysosomes, and T3, T4, diiodotyrosine and monoiodotyrosine are liberated into the cytoplasm. The iodinated tyrosines, diiodotyrosine or monoiodotyrosine, are deiodinated by a microsomal iodotyrosine deiodinase. This enzyme does not attack iodinated thyronines T4 and T3 that passes into the circulation. The iodine liberated by deiodination of monoiodotyrosine and diiodotyrosine is reutilized in the gland and normally provides about twice as much iodine as the iodine pump, that is sodium iodide symporter, does. So T4 and T3 are relatively lipophilic, thus their free forms in the plasma are in equilibrium with a much larger pool of protein bound thyroid hormones in the plasma and in tissues. The plasma proteins that bind thyroid hormones are albumin, a prealbumin called transthyretin, formerly called thyroxine binding prealbumin, and a globulin, thyroxine binding globulin or TBG. Of these three proteins, albumin has the largest capacity to bind T4 and thyroxine binding globulin has the smallest capacity. But the affinities of the proteins for T4 are such that most of the circulating T4 is bound to thyroxine binding globulin. In other words, the affinity to bind T4 is highest in thyroxine binding globulin. Normally 99.98% of the T4 in plasma is in bound form, and 99.8% of the T3 in plasma is in bound form. So biologic half-life of T4 is long, about 6 to 7 days, whereas T3 has a shorter half-life than T4, but its action on the tissues is much more rapid. So this is all about, not all about, this is the synthesis and transport of thyroid hormone. In next class we will discuss about the mechanism of action and hypo and hyperthyroidism. Thank you.